Afternoon to all the Bi.P.C second year students. So here we are in our last session of our IPE. So let's just look into the most important very short answer type of questions, session 2. In session 1 we have dealt with complete VSAQs of the unit plant physiology, and now we are going to cover up the rest chapters. Fine, right? So first let us start with microbiology and the chapter bacteria. If you look into the first question, what is conjugation, who discovered it, and in which organisms you can see this. Conjugation is nothing but, first you have to define what is conjugation, and then you have to say in which it is discovered and who are the scientists who has driven out this conjugation. First, conjugation is the process in which two bacterial cells will exchange the DNA through direct contact. By what? You will have two different strains of bacteria. F plus strain will have F factor in the plasmid DNA. It will be transferring the genetic material to F minus strain. Fine, right? F plus will be the donor and F minus strain will be the recipient. So this direct contact is established through sex pilus or sex pili. Fine, right? This involves a donor cell transferring its genetic material to the recipient cell. This is discovered by two scientists, that is Liderberg and Tatum, and in what model organism? Escherichia coli model organism. Next, what is plasmid and what is its significance? So here we need to look into the plasmid DNA. Plasmid DNA, here you see what is plasmid, what is its significance. Plasmid and its significance. A plasmid is a small circular double-stranded DNA. Yes, right. You can also define plasmid DNA as the extra chromosomal DNA, okay, or non-genomic DNA as well. You can also define this as non-genomic or extra chromosomal DNA. Non-genomic or extra chromosomal DNA. You can also name it that double-stranded DNA molecule which is found in the bacterial cells. This separates from the chromosomal DNA. It contains genes which are responsible to give a specific phenotypic trait, and this provides antibiotic resistance as well. Antibiotic resistance to many toxins protection. Plasmids are significant in biotechnology as vectors. This is also important for vectors. These are used as vectors for gene transfer and genetic engineering. Next question, if you see, what is genophore? Okay, what is genophore? Genophore is nothing but, this is a bacterial genetic material which is also called the bacterial chromosome. It's a bacterial genetic material also addressed as bacterial chromosome. It consists of single circular double-stranded DNA molecule. It carries all the essential genes of bacterial cell. It carries all the essential genes for bacterial cell function and its replication. This is about genophore, plasmid DNA and conjugation. Three major concepts. Many a times VSAQs are picked from these concepts. Now last two questions of this chapter, if you see, what are pleomorphic bacteria? Give an example. Pleomorphic, the name itself is saying it will exhibit multiple structural features. Pleomorphic bacteria. They can change their shape depending on environmental and nutritional conditions. Okay, it depends on many environmental and nutritional conditions. They lack a rigid cell wall will be absent, and this will allow variable morphology. Different types of morphological structures are being seen. Example, if you see, Mycoplasma species. Mycoplasma species are the examples. Now, name the bacteria which cause plant diseases like blight of rice and citrus canker. Okay, in answer, Xanthomonas oryzae will cause blight of rice, and Xanthomonas aconopodis, this will cause citrus canker disease. Both pathogenic bacteria they affect economic importance of the crops. Fine. This is about the short answer type of question concepts that are picked from bacteria. Next, if you see virus, what is lysozyme and what is its function? Okay, lysozyme is a hydrolytic enzyme, viral enzyme. Okay, lysozyme is a hydrolytic viral enzyme. This is synthesized inside the host cell. This will help in breaking the bacterial cell wall. Lysozyme will help in breaking the bacterial cell wall during viral infection. This facilitates the release of new virus particles like virions. Fine. This is about lysozyme enzyme. In lysogenic cycle, you can observe that feature. How the new virions will be released from the virus when it is infecting any host cell. What is the type of T4 phase? What is its genetic material? Asked multiple times in many years. Here you see T4 phase has tadpole shaped structure. It has tadpole shaped structure. Its genetic material is double stranded DNA. Genetic material of this is double stranded DNA. It infects E. coli bacteria and is used in phase studies. Okay, this will infect Escherichia coli bacteria and this is used in phase studies. What is prophase? Next is, what is prophase? A prophase is viral DNA. This is integrated into the bacterial chromosome. Once the viral DNA is integrated into the bacterial chromosome, then the term you address it as what? Prophase. It remains dormant without destroying the host cell. Under certain conditions, it can initiate the lytic cycle as well. So, whenever you are going to these two concepts about bacteria and viruses, in bacteria about plasmid DNA, that is very important, and you have to be clear on the note of lytic and lysogenic cycle. And if you see viruses, in viruses you have to be clear on lytic and lysogenic cycles, because the questions, both short answer type of questions might be, VSAQs might be, have arisen multiple times from lytic and lysogenic cycles of viruses. Clear with this answer, ma? And then mention the differences between virulent phases and temperate phases. This question also is in connection with lytic and lysogenic cycle. So, here you see virulent phases and temperate phases. This causes lysis of host cell after infection, whereas temperate phases, they do not cause any immediate lysis. No immediate cell lysis will happen in the when temperate phases will cause infection. Prophase is absent here, whereas here prophase is present in the host DNA. This will follow lytic cycle. Virulent phases will follow lytic cycle and temperate phases will follow lysogenic cycle. For example, lambda phase. Virulent phases will follow lytic cycle, for example, T even phases. Next, principles of inheritance and variation. Explain the terms phenotype and genotype. Phenotype and genotype. Observed in many years, if you see 2024, 2022, 17, 18 and 14. These many years, multiple times asked questions on genetics. On genetics, you can expect short answers, very short answer type of questions. So many questions are picked from principles of inheritance and molecular basis of inheritance. Same level of importance is given for your NEET as well. You can easily score up to 20 to 25 marks easily on the minimum base I am saying here. Easily you can score from genetics. So very important, you have to go through these concepts. So it is on the phenotype and genotype. Phenotype will refer to what? External morphological features or traits of an organism, whereas genotype is genetic constitutional makeup of an organism. Genotype, both homozygous and heterozygous for dominant traits will be there. For example, dominant tall is the trait, tallness is a trait, dominant trait. Two genotypes can be applicable for this according to the Mendel's pattern of inheritance. It can be homozygous tall or heterozygous tall. Here phenotype results from the interaction between genotype and the environment. Who proposed chromosomal theory of inheritance? Who has proposed this? Asked in multiple years like 2023, 22 and 2020 as well. Here we go with this. The chromosomal theory of inheritance, this is first proposed by Sutton and Boveri. Sutton and Boveri has proposed chromosomal theory of inheritance. Okay, Sutton and Boveri has proposed chromosomal theory of inheritance. This will explain that genes are located on the chromosomes. Okay, and the behavior of chromosomes during meiosis is parallelly with the segregation and assortment of the genes. Okay, the segregation of the genes is in parallel movement with chromosomes. Chromosomal behavior is coinciding with the segregation of genes is what finalized by Sutton and Boveri in chromosomal theory of inheritance. Next is, what is point mutation? What is point mutation? Give an example. Asked in multiple years like 2023, 22, 2019 and 2018. In multiple years this question is asked. So if you see this question, point mutation will occur when a single base pair of DNA is altered by substitution, insertion and deletion. Okay, here single base pair of DNA, this is altered by substitution, insertion and deletion. It will affect only one gene and may change the function of the entire protein. The complete function of a protein will be changed here. Example, if you see, sickle cell anemia is the example. Fine. Next, define true breeding and mention its significance. Asked in 2022 and 2016 if you see. So here you look into true breeding will refer to continuous self-pollination that maintains stable genotype and phenotype across the generations. Across generations, it will maintain stable genotype and phenotype. This helps to produce genetically uniform offsprings with predictable traits. And then this is useful for determining genotype of plants during test cross as well. So whenever you are mentioning the accurate points with respect to the concept, don't forget to highlight them or underline the major terms which are involved in the question. Fine. And then, what is the cross between F1 progeny and homozygous recessive parent called? How is it used? Again, if you can see, multiple times asked question. The cross between F1 progeny and homozygous recessive parent is observed as test cross. It helps identify the unknown genes, unknown genotype of F1 individual. Offspring ratio will reveal whether the F1 is homozygous or heterozygous. F1 is whether homozygous or heterozygous, the offspring ratio will reveal this. Next, what will be the phenotypic ratio in offsprings obtained from the following crosses? If you see the phenotypic ratio, when heterozygous dominant trait is crossed with homozygous recessive trait, what will be the phenotypic ratio? Look at the first question. It will be very impressive if you can draw a checkerboard and explain this. If you look into this, one heterozygous dominant and homozygous recessive, you will get two heterozygous dominant and two homozygous recessive. 1:1 ratio you will get. Homozygous dominant and homozygous recessive. When they are crossed, if you see, all offsprings will be tall itself. All offsprings will be heterozygous tall. Next, two heterozygous traits have crossed. In that case, 3 will be tall and 1 is dwarf. 3 will be dominant and 1 will be recessive. Clear? Next question you see. In garden P, the gene capital T for tall is dominant over the allele of dwarf. So, tall and dwarf ratio if you observe, all offsprings will be tall itself, heterozygous tall. All offsprings will be heterozygous tall itself. And here offsprings, when two tall species have crossed heterozygous tall, you will get 3:1. This is being observed in Mendel's monohybrid cross, where he has driven out Mendel's law of dominance. Next, you look into the chapter that is molecular basis of inheritance, and the first question is on initiator codon or the start codon. What is the function of AUG? Asked multiple times, and question on AUG you can also see in the your NEET and MCET objective questions as well. So here if you observe, AUG acts as start or initiator codon in mRNA, messenger RNA. It begins the process of protein synthesis. Okay, it will begin the process of protein synthesis. It also codes for amino acid methionine. AUG codes for methionine. Fine, right? Next, in a typical DNA molecule, the proportion of thymine is 30% of N bases, nitrogen bases. Find out the percentage of nitrogen bases, total nitrogen bases percentage he is asking. So we know, according to Chargaff's rule, the amount of purines will be equal to the amount of pyrimidines, right? So here if you see, number of adenine, amount of adenine will be equal to the amount of thymine, amount of cytosine will be equal to the amount of guanine. Fine. And here we go. First, you see, like thymine is 30%, which means adenine also will be of 30%. Right now, in that case, if you assume 100% of nitrogen bases in a double stranded DNA, where you can see 60% itself is occupied by adenine and thymine, from rest 40%, cytosine will be of 20% and guanine will be of 20%. Understood everyone? Any doubt anywhere ma? Is this clear till here? Yeah. So in that ways you can solve this question. This type of questions are also being asked in your MCET and NEET. Okay. Fine, right. Next question you observe. What are the components of a nucleotide? Components of a nucleotide. Nitrogen base. It can be either purine or pyrimidine. Give out the nitrogen bases which are present in the nucleotide. It can be purine or pyrimidine. If it is DNA, deoxyribonucleic acid, then the nitrogen bases can be adenine, thymine, guanine, cytosine. If it is RNA, then it can be adenine, uracil, guanine and cytosine. So this will be a pentose sugar, which is a 5-carbon compound. It can be ribose in RNA and deoxyribose in DNA, where at the second carbon there will not be oxygen group, free O group. A phosphate group will link the nucleotides together, where the phosphate group is present in the nucleotide at the 5th carbon of the sugar, the phosphate group is binded. Clear? Now the question is about stop codons. Write the codons. About stop codons you can see asked in multiple times, like about initiator codons and start codons, multiple times we got. So a stop codon is nothing but this will signal the termination of protein synthesis, and 3 stop codons are present. You can see UAA, UAG and UGA. These are the 3 stop codons. They do not code for any amino acids. Okay, they do not code for any amino acids. And then what are the components of transcription unit? What are the components of the transcription unit? Here you see in the promoter. Promoter, it will initiate the transcription. Structural gene, they will encode for RNA sequence, and terminator, this will mark the end of the transcription. Fine. These are the components of a transcription unit, starting from promoter followed by structural gene and then ends by terminator. Then what is the difference between exons and introns? Difference between exons and introns. Asked in multiple years like 2023, 22 and 2018. Exon is nothing but this is a coding sequence which is expressed, coding sequence or expressed regions of genes are exons. Introns are non-coding sequences. In simple terms, you can remember this way beta. Exons are expressed sequences, introns are inactive sequences. That way you can remember. Expressed sequences of gene sequences in a DNA. Introns are inactive regions. Next, they appear in mature or processed RNA, and here introns, they are removed during RNA processing. Exons contribute to protein synthesis. Introns, they will not appear in the final mRNA. Introns will not be appeared in the final mRNA. Next, the proportion of nucleotides in given nucleic acid are adenine is how much percent ma? 18%. Guanine is 30%. Cytosine 42 and uracil 10%. What is the name of name the nucleic acid and mention the number of strands. The presence of uracil itself, you forget about this percentages on and all, but the presence of uracil itself is telling that the genetic material or the nucleic acid present here is RNA, not DNA. Unequal base ratios confirm that it is single stranded. If it is double stranded, then equal pairing will happen, right? So it is a single stranded RNA because it has unequal pairing. Hence the molecule is single stranded RNA. Molecule here is single stranded RNA. Next, given below is the sequence of coding strand of DNA in transcription unit. 5 prime to 3 prime we have. Write the sequence. It's complementary strand of the mRNA. Here we go. Complementary DNA strand for the above mentioned. If it is from 5 prime to 3 prime, you have to start it from 5 prime to 3 prime because it is the synthesis is always from 5 prime to 3 prime. Yeah. The complementary strand for the above given strand is this. If you can see at 3 prime region which starts from 5 prime here, adenine is there, the complementary strands will be thymine. Fine, the complementary DNA strand is this. Now, as the coding strand is mentioned in the question, mRNA will be same as that of coding strand, except in the place of thymine in mRNA sequence, uracil will be there. So what you have to do, it is very simple. Whenever the coding strand is given in the question, not even in IPE, in MCET or NEET, whenever in your question coding strand is given and they are asking you for mRNA, don't change anything from 5 prime to 3 prime. Everything will be as it is, all the nitrogen bases. Only one change, that in the place of thymine you will be using up the uracil. That is the only change that we make up. The mRNA sequence is complementary to the DNA template strand, replacing thymine with uracil. Clear till here? What is meant by capping and tailing? What is meant by capping and tailing? Asked in March 2021 to 2016, multiple years asked question. Capping, if you see, addition of modified guanosine triphosphate. Modified guanosine triphosphate which is added at 5 prime end of the mRNA strand. Tailing, if you see, poly A, multiple adenine sequences like 200 to 300 residues are being added at the 3 prime end of heteronuclear RNA. Both processes, 5 prime capping and 3 prime tailing, both the processes will protect mRNA and this will make ready for translation process. Okay. Next, distinguish between heterochromatin and euchromatin. Which of the two will be transcriptionally active? We know that what is transcriptionally active? Euchromatin, which is lightly stained. Okay. Structure, if you see, heterochromatin is densely packed structure, euchromatin will be loosely packed. Okay. And staining procedure, heterochromatin is darkly stained, euchromatin will be lightly stained. Gene activity, this is transcriptionally inactive and euchromatin is transcriptionally active. Heterochromatin, this is found in the nuclear membrane, this is found in the inner region of the nucleus. Replication timing, this replicates late during S phase and euchromatin replicates early during the S phase. Till here everything is fine, right? This is the clear differentiation between heterochromatin and euchromatin. What is the difference between template strand and coding strand of the DNA? Template and coding strand of the DNA. Look into the answer. Template strand runs from 3' to 5' direction. This serves as template strand for mRNA. Okay, this will be the template strand for mRNA. Coding strand will run from 5' to 3' direction and coding strand will decide the position of promoter and terminator. The 5' region of the coding strand will be the promoter region and 3' end of the coding strand will be the terminator region. This has the same sequence as that of RNA, except in the place of thymine it will be replaced by uracil. Clear? The two strands are complementary to each other. Write any two differences between DNA and RNA. Any two differences. You can consider sugar and nitrogen base, okay, or you can consider the single stranded, double stranded forms as well. Here I have mentioned sugar and nitrogen base. You can see here it is deoxyribose sugar, ribose sugar. Nitrogen base, here it will be thymine, and in the place of thymine in RNA uracil will be present. Look at the next question. Alfred Hershey and Martha Chase proved that DNA is the genetic material. Who proved or who proposed that DNA is the first genetic material? It is Hershey and Chase experiment who unequivocally proved that DNA is the genetic material. They conducted experiments on what organisms? On bacteriophages. On bacteriophages they have conducted the experiments. Their experiments confirmed that DNA, not protein, this will enter the bacterial cells to transmit the heredity. Next, what is the function of DNA polymerase? DNA polymerase function. DNA polymerase will catalyze the polymerization of nucleotides. This will catalyze the polymerization of nucleotides during DNA replication. This is from 5' to 3' and it ensures high fidelity in DNA synthesis. It also helps to unwind separate strands by breaking what? Hydrogen bonds during replication. This is about DNA polymerase, and DNA polymerase is the most efficient enzyme that you can see during replication. Fine. When you look into the complete enzymatic machinery of replication, it is DNA polymerase which conducts the synthesis of new strand most effectively. Next, questions from biotechnology, principles and processes. What is the full form of PCR? How is it useful in biotechnology? Multiple times asked question. Polymerase chain reaction is very important for your objective exam also. PCR is polymerase chain reaction. What is the main activity that we do here? Amplification of DNA sequences in vitro. Okay, amplification of DNA sequences in vitro. This will help in what processes? Gene cloning, DNA fingerprinting and diagnostics. This will help in all these three processes: gene cloning, DNA fingerprinting and diagnostics. Next, what is downstream processing? Okay, downstream processing asked in multiple years like 2023, 2017, 2016 and 2014. We look into this process. Downstreaming will refer to purification and recovery of biotechnological products. This includes steps like separation, purification and formulation. This ensures production of commercially pure product which is suitable for the use. Fine, downstream processing is very important, ma. Differentiate between exonucleases and endonucleases, again important concept from your restriction enzymes. Look into it. Exonucleases will remove the nucleotide sequences at the ends, whereas endonucleases will target the specific internal sites. This acts progressively from either 5' or 3' ends. This will recognize the specific sequences. Here comes the palindromic sequences into picture. Example of exonucleases, if you see exonuclease 1, endonucleases, example restriction endonuclease, that is EcoR1. How do one visualize DNA on an agarose gel? DNA fragments are stained with ethidium bromide. These are stained with ethidium bromide after electrophoresis. Under UV light, the stained DNA will emit orange fluorescence, and the bands indicate different fragment sizes for analysis. What is EcoR1? How does it function? EcoR1 is restriction endonucleases enzyme which is obtained from Escherichia coli. This is obtained from E. coli. Fine. It recognizes palindromic sequences. Palindromic sequences will be from 5' to 3' direction, and this will cut in between guanine and adenine. This cleavage creates sticky ends. Sticky ends will be created which is useful in gene cloning. Okay. Name any two artificially restricted plasmids. Restructured plasmids, artificially restructured plasmids. Two examples here you give, the answer is depending on the examples only as they have asked. PBR 322 and PUC 19. Next, the question is on biotechnology and palindromic sequences. Look at this first question. Biotechnology is the branch of science. It will use living organisms or biological systems for developing useful products. It will apply scientific and engineering principles to produce goods that will improve the human welfare. It includes genetic manipulation, fermentation and tissue culture techniques as well. What are the palindromic sequences? Palindromic sequence in DNA, it will consist of inverted repeats that read the same from 5' to 3' directions on both the strands. These sites will be recognized by restriction enzymes for cutting the DNA. Example, if you see from 5' to 3', GAATTC will be CTTAA, from 3' to 5'. These are the palindromic sequences. Molecular scissors. Molecular scissors nothing but restriction endonucleases. They will cut DNA only at the specific sites. They will target the internal specific sites. Fine. They are obtained from bacteria such as Salmonella typhimurium and Escherichia coli. These enzymes are essential tools in genetic engineering. Biotechnology and its applications, if you see, what is GEAC and what are its objectives? What are the objectives of this? Observed in many years like 24, 2023, 22 and 2017. GEAC is nothing but Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. This will regulate and approve genetically modified organisms for research and release. It will ensure both safety and ethical use of genetically modified technology in India. Fine, right? What is Green Revolution? Who is regarded as father of Green Revolution? It is obviously Norman Borlaug, who is the father of Green Revolution. And what is green revolution for? To increase the agriculture production through improved crop varieties and breeding. Those 2 points you mentioned, that will be enough. Next, for which variety of Indian rice has patent been filed by USA company? It is for Basmati rice, which is an Indian variety. The patent right has been filed. That is the major point will be looked for. Name the nematode that infects roots of tobacco plant, and name the strategy name the strategy adopted to prevent this infection. The nematode just a second. The nematode Melodogyne incognita will infect the roots of tobacco plant. It will infect the roots of tobacco plant. The control strategy is used as RNA interference, that is RNAi, silences specific genes in the pest and preventing infection. Just a second. Here we go. Can a disease be detected before its symptoms appear? Explain the principles involved for the disease. Yes, a disease can be detected in early by amplifying pathogen DNA using PCR technique. PCR will produce millions of copies of target DNA for early detection, and the techniques like PCR and ELISA are used for diagnosis before visible symptoms appear itself. Next, strategies of enhancement in food production from this chapter. Crygenes, very important. Different crygenes and pest controlling by proteins encoded by these genes. If you look into crygenes, which different types of crygenes, cry1AC, cry2AB, crylab, these are different genes. They are for controlling cotton bollworms and corn borer pests. Next, two species which are produced by crossbreeding. In sugarcane, if you see Saccharum barberi and Saccharum officinarum were crossed, this will produce what type of qualities or traits? The hybrid combined high yield and disease resistant traits, and the cross improved sugar content and adaptability to tropical climates. Two examples of fungi used in single cell protein production. Candida utilis, that is Torula yeast, Saccharomyces cerevisiae, Baker's yeast, both are used in single cell protein production for nutrition. Which part of plant is best suited for making virus free plants and why? So here apical and axillary meristems, they are best suitable for virus free plants. These regions are free for viral infection and they can be grown in vitro using tissue culture to produce virus free plants. What is meant by hidden hunger? This refers to deficiency of essential micronutrients, and this leads to diseases and disabilities despite adequate calorie intake. And the main cause is inability to afford and access nutrient rich foods. Two examples for wheat varieties, Sonalika and Kalyan Sona. These are the two varieties of wheat. Totipotency. What is totipotency? Any cell, a single cell which has the capacity to regenerate the entire plant. That phenomena is known as totipotency. And what is an explant? Any plant tissue or organ grown in sterile nutrient medium under controlled conditions is known as explant. What is meant by biofortification? Biofortification is nothing but development of nutrient rich crops through breeding or biotechnology. It will increase the levels of proteins, vitamins, minerals in crops. Fine, right? The goal is to combat malnutrition and hidden hunger. What is meant by germplasm collection? Germplasm collection will involve gathering plant seeds with diverse genetic traits. It will preserve distinct alleles of all genes in a species. Such collections are valuable for crop improvement and conservation. Last question of strategies of food production. Here we go. Define micropropagation and somaclones. Rapid production of plants using tissue culture technique is micropropagation. Somaclones, plants produced through micropropagation that are genetically identical to that of parent. Microbes in human welfare, scientists who credited for showing the role of penicillin as antibiotic. The discovery and development of penicillin are credited to Alexander Fleming, Ernst Chain and Howard Florey. Fleming first discovered penicillin in the year 1928 and later they have developed the therapeutic potential. Swiss cheese is by Propionibacterium shermani and large amount of carbon dioxide will be released, that is why big holes are formed on the Swiss cheese. Next, nucleopolyhedroviruses begin being used for nowadays. What type or what is nucleopolyhedroviruses? They are used as biological control agents against insect pests. They have narrow spectrum of insecticidal applications and it is species specific. Next is about fermenters. Fermenters are large bioreactor vessels. They are used for growing microorganisms on an industrial scale. They provide optimal conditions will be provided for temperature, pH, aeration and nutrients. They are essential for mass production of enzymes, antibiotics and beverages. Name a microbe used for statin production. Okay. And how do statins lower blood cholesterol levels? Microbe used for statin production is Monascus purpureus. Here statins will inhibit HMG-CoA reductase, the key enzyme which is used in cholesterol biosynthesis. Next, this reduces blood cholesterol levels which prevents cardiovascular diseases as well. What is the chemical nature of biogas? Name the bacteria which is involved in the production of biogas. Biogas is mainly composed of methane, carbon dioxide and hydrogen. Fine. It is produced through anaerobic decomposition of organic matter and the key bacterium involved is methanobacterium. This is the key bacterium which is involved for the production. Fine. Two industrial important enzymes. Many enzymes you can take like amylases, lipases, proteases, streptokinase. Industrially important enzymes they are. Next, last questions of today's session. Name the immunosuppressive agent and where is this obtained? Cyclosporin A is the immunosuppressive agent and it is obtained from the fungus Trichoderma polysporum. Name two genetically modified crops, Bt cotton and Bt brinjal. They have cry genes, cry1AC, cry2AB. This genes from Bacillus thuringiensis providing pest resistance. Clear? So, with this complete IPE questions of second year botany are dealt. If you are having any doubts on concept clarity or what type of concepts should be focused additionally, you can please drop messages in the chat box. I will be addressing your doubts. And at last, I would like to end up the session by saying that all the students remember this. Your IPE preparation is nowhere isolated with your objective preparation. Never have that in your mind. Make on a parallel track of preparation. Like for example, if you are planning or learning a concept, that concept you have to have a framework on how you are presenting that concept for subjective answer, and practice the objective related to that concept. Parallelly, your MSET and NEET preparation also will be done. Likewise, you plan your preparation, but then don't have an isolated IPE preparation, and after IPE preparation don't start for preparing for NEET and MSET. Don't waste your time in that way. So saying this, I would like to tell you all the very best for your exam, and that's all for IPE sessions.